Okay, today is the digestive system. Put this in slideshow mode. Come on, buddy. Okay, the digestive system is composed of what's called the alimentary canal or the gastrointestinal tract. It's a continuous tube that begins at the mouth and ends essentially at the rectum. Functions to digest and absorb nutrients, includes the mouth, pharynx, esophagus, stomach, small intestine, large intestine, all the way through the rectum. So we have the tract, which is that tube, and everything, the different regions of essentially the tube or the distended tube, which I consider the stomach to be. And then you have your accessory organs, those that lie kind of outside the GI tract, but communicate with it. They function to production of secretions that aid digestion and mastication of food. And they include things like the teeth, tongue, salivary glands, liver, gallbladder, and the pancreas. So here we have the entrance point of food into the mouth, and then where it terminates at the rectum. So we're going to start at the beginning and work through structure, function, and we'll look at some hormones. And then the next chapter is the chemical digestion, the enzymes and processes associated with digestion. Okay, so we have these particular processes. Ingestion is a term that refers to intake of food into the GI tract. Mechanical breakdown includes the specific kind of actions of mastication, which is chewing, churning and segmentation. We have gastric churning, we have haustal churning, gastric churning in the stomach, haustal churning in the large intestine, and segmentation that occurs in the small intestine. We have propulsion, which is the moving of food by muscular contraction. We have chemical digestion. That's how we chemically break down foods with enzymes. Absorption, which is the movement of nutrients from the GI tract to the blood or lymphatic systems. And defecation is the elimination of undigestible material. So starting at the mouth and working our way through, this is kind of a handy picture to refer back to periodically. I don't ask a lot of questions, from the previous slide. Basically, it would be maybe the definitions of these things, but that you can see how food gets taken into the mouth. That's where we ingest food, the only place. And we're going to move it down through the esophagus to the stomach, from the stomach to the small intestine, to the large intestine, and then eliminate whatever we want to, out the rectum. So we have mechanical breakdowns such as chewing, which is mastication, churning of the stomach, churning in the small and large intestine farther down, segmentation in the small intestine. We have forms of propulsion like swallowing and peristalsis. Absorption primarily occurs in the small intestine, but we absorb certain molecules in the stomach as well. The absorbed molecules will either enter the cardiovascular system or into the lymphatic system. So the only thing that goes in the lymphatic system are going to be building blocks of fats called chylomicrons. And then we follow the system down. Then we have defecation. So, right here, they'll have, at least in the big squares, ingestion, mechanical breakdown, digestion, propulsion, absorption, defecation, and where they occur. But it's kind of helpful to look at. Okay, so we have basically, kind of like when we looked at the trachea, we have 4 layers, of tissue. And this is the basic layers, but there's some modifications to these depending on the organ that you're in. Especially when it comes to the stomach, we're going to have 3 layers of muscle, not just 2. But we have these 4 basic layers of mucosa. But here we find simple columnar epithelium, submucosa that consists primarily of areolar connective tissue, and simple cuboidal epithelial tissue, because the entire system is lined with mucus, so that a lot of that mucus is coming, or the mucus is coming from the mucus glands of the submucosa. Submucosa contains blood vessels and lymphatic vessels, nerve plexuses that supply the GI tract, and mucus glands. All right, then we have radiating outward the muscularis layer. This is smooth muscle. The majority is composed of an inner circular and then an outer longitudinally running muscle fibers. So this circular muscle runs circumferentially around the tube, like the smooth muscle of a blood vessel. And then superficial to that are longitudinal muscles that run the length of the tube. And then we have the serosa, which is the outer connective tissue layer. And just like any outer connective tissue layer, helps secure the structure in place and secures the nerves and blood vessels and lymphatic structures that supply them. Okay, so let's start in the mouth of the oral cavity. Now, the primary tissue is stratified squamous epithelium, and the muscular tissue is skeletal muscle because that's under volitional control. The mouth is the point of intake of food. The tongue is where we have taste buds, propels food for degglutination or swallowing. And don't forget, this tongue is associated with speech and forming words. We have salivary glands that contain saliva, and saliva is mostly water, and it contains amylase, where we start carbohydrate digestion. Amylase starts breaking down a polysaccharide to a disaccharide, or starch to a disaccharide. The water helps moisten the food so it doesn't, it's easier to swallow, but it also provides an aqueous solution for molecules to be suspended. And the chemoreceptors of your tongue need to have these tastins dissolved in aqueous solution in water for them to interact, with the chemoreceptors of the taste buds. And then we have teeth for mastication, chewing, grinding, shred, or tearing of food. So here we have the oral cavity, stratified squamous epithelium in the oral cavity to the oropharynx and laryngopharynx, right? And we have the tongue, which is composed of skeletal muscle tissue. Okay, so let's start looking at teeth. We have incisors, canines, premolars, and molars. Incisors cut like a knife. Canines shred or tear. And premolars and molars grind. They have a broader flattened surface, not completely flat, but flatter. So here we have the incisors. We have 4 on the lower jaw, 4 in the upper jaw. And so if we start midline and move to, let's say, and here we're moving to the left, we have 2 incisors, a canine, 2 premolars or bicuspids, and 3 molars. Cut incisors, shred canines. See, and then you have these flatter surfaces to the premolars and molars. Those are grinders. You can also see the location of some of the ducts, some of the glands, the salivary glands. We have a parotid gland, which is superficial to the temporomandibular joint. We also have a sublingual glands that would be inferior to the tongue, and submandibulars that are inferior to the mandible. Okay, the mouth, pharynx, and esophagus. Right. Mastication is the mechanical breakdown of food by the teeth. Degglutination is swallowing. Okay. And then peristalsis is the radially symmetrical contraction and relaxation of muscles that propagate in a wave down a tube in a forward direction beginning in the esophagus. So what this means is that when you deglutinate, you swallow, the food is going to pass into the esophagus. And this is where we have a lot of smooth muscle to start to push that bolus of food that you just swallowed. That's a amount of food in one swallow. And it's going to propel it, squeezing behind it, pushing it down, like squeezing toothpaste out of the toothpaste tube. squeezing the food and moving it down to the stomach, and then from the stomach through the small intestine and large intestine. Okay, the glands. We have the parotid gland anterior to the ear and external to the masseter. It sits kind of on top of your temporomandibular joints, very superficial. Then we have a sublingual gland below the tongue and submandibular gland below the mandible. They function to produce secretions to dissolve food chemicals for taste, as I described before. Taste stints need to be dissolved in aqueous solution or water to interact with the chemoreceptors of your taste buds. It moistens food and compacts into a bolus and begins starch breakdown with the enzyme salivary amylase. So salivary amylase starts carbohydrate digestion. We start to break down that polysaccharide to a disaccharide. Also in salivary amylase, we do have a little bit of what's called lingual lipase, which means that it's just going to act on starting to break down some of the fats. The majority, and it'll say start, the majority of fat digestion is going to take place in the small intestine from the lipase produced by the pancreas. But I have to admit that yes, in saliva, there's a little bit of lingual lipase. Okay, so here we see the oral cavity, and then we see the teeth, the upper row, the lower row. We have the parotid gland that is superficial anterior to the ear here. We have the sublinguals and submandibular glands. Then we have the pharynx and the esophagus. So the pharynx is histologically similar to the oral cavity. Remember, both the oropharynx and laryngopharynx are lined with stratified squamous epithelium. Right, then we enter the esophagus. The esophagus propels food to the stomach, and this is where peristalsis begins. At the entrance point of food, we have the majority of this esophagus is lined with stratified squamous epithelium. The muscle changed from skeletal muscle to smooth muscle. That sits under the control of the autonomic nervous system. And the outer layer is termed adventitia because we are in the thoracic cavity. Remember, the outer connective tissue layers are technically referred to, as adventitia. And then once we get into the abdominal pelvic cavity, it's called serosum. I'm not going to ask you to distinguish those two things, but if you keep wondering what the difference in the terms is, it's the outer connective tissue layer. If the organs in the thoracic cavity, we call it adventitia. If we're now in the abdominal pelvic cavity, we call it a serosum. Okay, so now we're in the stomach. The rugae, or the folds that you see in the stomach when the stomach is rather empty, are called rugae. And once they allow the stomach to distend and stretch, and accommodate more food, so they help increase the volume of the stomach. We have an entrance point and an exit point. We have two sphincters. From the esophagus to the stomach is the cardiac or lower esophageal sphincter. That has to relax and contract. Relaxes when food passes through the esophagus into the stomach, and generally will contract once the food enters the stomach because you're going to secrete a lot of hydrochloric acid, and that churning that occurs there, we don't want the hydrochloric acid to kind of regurgitate back up into the esophagus. So that sphincter works well to prevent that. And then between the stomach and entering the small intestine, we have the pyloric sphincter, and that regulates the entry of food, or what's called chyme at this point, into the small intestine. You don't want to have that door or window wide open. So as the stomach is secreting all these different acids to the surface of the food that you're eating, you don't want it to just all enter the small intestine all at once because has a very low pH. So you need to have small amounts, be delivered from the stomach into the small intestine. The small intestine can then modify it, try to elevate the pH. Hormones are going to be liberated to, open up the sphincter ability to let pancreatic juices and often bile to enter into the small intestine to facilitate chemical breakdown. So we have these two sphincters at the beginning, entrance point to the stomach, and at the exit point. And so here we have a picture of the stomach. We have the lower esophageal or cardiac sphincter here where the esophagus meets the stomach. And then leaving the stomach, we have the pyloric sphincter to the bottom that regulates the amount of chyme from the stomach into the small intestinal duodenum. Chyme is just what we call that fluid now that's been being churned around in the stomach. It's called chyme. Keep in mind, we also have 3 layers of muscle here, which in the muscular layer that is different from the rest of the digestive system. So now's a good time to look at it because it identifies your oblique layer, your circular, and your longitudinal layers. 3 layers. Okay, so the stomach and its microanatomy. We have the mucosa, of course, contains lots of mucus glands. Secretes mucus for autoprotection from acidic gastric juices. Right, a lot of mucus glands are down in the submucosa as well. We have parietal cells that secrete hydrochloric acid. Hydrochloric acid serves a couple different functions. One is to stimulate the conversion of pepsinogen, which is the inactive form of pepsin, and pepsin begins protein digestion. It begins protein digestion, breaking down a polypeptide to a dipeptide. Hydrochloric acid also helps kill bacteria. And it stimulates intrinsic factor, or not the hydrochloric acid, but the parietal cells release intrinsic factor. And remember, the intrinsic factor is required for B12 absorption. We also have chief cells, and chief cells produce the pepsinogen. The enteroendocrine cells, we've often referred to as G cells, secrete gastrin, which is a hormone that stimulates the further production of gastric juices. So what does that mean? It means that when you swallow food, it starts to change the pH of the stomach, and the stomach senses that. It goes, okay, we've now ingested food. The pH is elevating because we're no longer just some hydrochloric acid there. And it goes, okay, we need to start secreting more mucus, hydrochloric acid, intrinsic factor, secrete more pepsinogen so we can convert it to pepsin. The submucosa is about the same, a lot of areolar connective tissue, simple cuboidal epithelial tissue. The muscularis layer expands to 3 layers to churn food and for peristalsis. It has an inner oblique, that's the extra layer, and then it has a circular and then a superficial outer longitudinal muscle layer. The outer connective tissue layer, now called the serosa, is the same, areolar connective tissue, same functions. Stomach digestion, mostly in holding area, for food. It aids in mechanical breakdown by producing what's called gastric churning. It's one of the physical ways, mechanical ways we digest food in the stomach. Most of it is going to be the pepsin and the actions of hydrochloric acid, denaturing a lot of the hydrogen bonds of these large proteins. But gastric churning allows material to be kind of rolled back and forth in the stomach to keep increasing the surface area, so it will facilitate breaking down those foods. Chemical digestion, proteins by pepsin. We had the hormone gastrin stimulates hydrochloric acid release and thus the conversion of pepsinogen to pepsin and gastric churning. We have absorption of lipid-soluble molecules like alcohol, aspirin, and drugs. And the only essential for life function here is the secretion of that intrinsic factor for absorption of B12. You need to have B12. You need to have B12. So remember that intrinsic factor is required for B12 absorption. So B12 is needed for red blood cells to mature. We use other, there's B5 and B6 and other ones, but we definitely need B12. Lack of intrinsic factor can cause pernicious anemia, and it's generally treated with B12 injections. chyme is a semi-fluid mass of partly digested food that is expelled from the stomach. So once it's mixing with hydrochloric acid and these different gastric secretions, we call that chyme. Okay, let's move on to the small intestine. So we're kind of a conveyor belt working its way from where food enters in the mouth, and we're going all the way through. And every time we pass through a different region, we're going to talk about, the histology and the functions there. So the small intestine is the major site of digestion, absorption of nutrients and water. Right? So it's a long, narrow tube, and thus facilitating this process. Once we get to the large intestine, we are pretty much done, with few exceptions for absorbing nutrients and chemically breaking down anything. Now we're just pretty much last ditch effort. We're absorbing some molecules, we're storing and forming feces. That's it. Okay, so we have three regions: the duodenum, the jejunum, and the ileum. The duodenum contains the hepatopancreatic sphincter, or the sphincter of Oddi, right, that regulates the passage of bile and pancreatic secretions from the bile duct system and from the pancreatic duct system, where they merge and they have a common entrance point into the duodenum. The jejunum contains cells called the brush border cells, that produce brush border enzymes. And these will be some enzymes that complete protein and carbohydrate digestion primarily. And then the ileum contains bands of muscle called and terminates at the ileocecal valve at the large intestine. And that ileocecal valve functions to sort of like the cardiac sphincter. It prevents backflow. It prevents material from the stomach regurgitating kind of back into the esophagus. The ileocecal valve helps prevent bacteria and fecal matter in the large intestine kind of regurgitating into backwards, into the small intestine. So here we have the stomach. The duodenum is the first region of the small intestine. It's rather brief. Then we have the jejunum and the ileum, and then that merges, with the large intestine at the ileocecal valve. Small intestine includes a mucosa, and that is composed of simple columnar epithelial tissue. We have lots of intestinal glands there. We have goblet cells embedded, between the columnar cells. We have the Peyer's patches that kind of span the mucosa to the submucosa, the lacteals, and of course capillary beds within it. And these most of these can be seen on any model. Okay. So the intestinal glands secrete digestive enzymes to complete digestion. Goblet cells secrete alkaline mucus to neutralize the acidic chyme from the stomach. We are going to line the entire system with mucus so that we don't burn holes through the walls, because these secretions can be very acidic, or the food can be very acidic. But remember, it's also a first line of defense for pathogens. Right, we have the Peyer's patches, those lymph nodes of the small intestine, the lacteals, that are part of the lymphatic system that can transport dietary fats. And then, of course, we have the capillaries within there to help absorb certain molecules from digestion and transport them to the liver. We have the mucosa with areolar connective tissue and mucus glands. So wherever you have glands, you have simple cuboidal epithelial system, simple cuboidal epithelium. And then we have the muscularis layer. It is smooth muscle, of course. And we go back to the inner circular and outer longitudinal layers of muscle. And then the outer connective tissue layer is the serosa. It's all the same. Now, some things, and there are some modifications of the small intestine that help with absorption of molecules. And one of them are the circular folds called the plicae circularis. So in the mucosa and submucosa, it folds into the lumen of the small intestine, and it helps slow down the movement of chyme and also increases the surface area. So unlike the rugae, which are folds of the stomach mucosa, because the the organ is collapsed. It's not filled with fluid, which will stretch and those rugae disappear, kind of like blowing up a balloon, as the stomach fills. But the circular folds of the plicae circularis in the small intestine are folds that are circumferential, go around the entire circumference, not like the length of the small intestine. And therefore, they do not disappear upon distension. And so, you not only have these folds inward, you're going to have on the surface of those folds villi and microvilli. And all these together help increase the surface area for maximal absorption. The villi are finger-like mucosal projections that increase the surface area. In the center, we have lacteals and lots of capillaries. And then we can have microvilli, or what's called the brush border, farther down in the small intestine, where we have those special cells that help secrete enzymes to complete carbohydrate and protein digestion. We'll look at those steps and those particular enzymes in more detail in the next chapter. All right, so here we have the small intestine. I like to use slides like this histologically in lab, where you can see the mucosa, submucosa, and then the muscular layers, and then whatever is superficial to the muscle layers there would have to be the serosa, or the connective tissue layer. So over to the right-hand side, it kind of brackets it, so you can see the villi, and they're cut open. They look like little fingers sticking up, but each one is one villi, and starting at the apex of that extension, all the way down to the bottom of like a darker purple staining region is your mucosa. And each villi on the surface is a layer of simple columnar epithelial tissue. You can see at certain regions, you can see some little goblet cells in there. In the center of those villi would be capillaries and a lacteal. And then as you travel down to the submucosa, you'll have areolar connective tissue, and you can actually see the glands on end. And you can see some what would be simple cuboidal epithelial tissue forming those mucus glands. And then you get to the muscularis layer. So you see that over to the right-hand side. The majority, about 75% of that layer, is going to be the inner circular muscularis layer. And so the layer that's closest to the submucosa, that's the circular muscle layer. And then to the outer region, about a quarter of it, the outer quarter, would be the longitudinal muscle layer. And then you have the serosa, the outer connective tissue layer. So small intestine digestion requires slow delivery of chyme from the stomach into the pyloric sphincter, or through the pyloric sphincter into the small intestine, so we can modify the pH. It has a very low pH. It could be like 2.5 to 3.0, somewhere through 3.5. And that's a pretty strong acid. So we need to neutralize that by allowing the bicarbonate that's formed in the pancreas to enter into the small intestine, elevate the pH. And that's for two reasons. You don't want to burn holes to the walls. Otherwise, you're going to create an ulcer, not like a duodenal ulcer. But you also remember that the enzymes from the pancreas are proteins, and proteins denature, break their hydrogen bonds when you have a low pH environment. So we need to elevate that pH so we don't burn and break up all our, denature all our proteins, all our protein enzymes from the pancreas before they can act and break these molecules down. If we don't break them down, we won't absorb them. You're just going to poop them right out. So it takes time to modify the acidity. The bicarbonate from the pancreas neutralizes the acid. And that serves, as I said, multiple functions. Okay, as we move down the small intestine, we come across those brush border cells that produce the brush border enzymes. We have maltase, sucrase, and lactase that will be secreted that act on maltose, sucrose, lactose, our disaccharides, to break them down into monosaccharides. We also secrete dipeptidase, and dipeptidase breaks down dipeptides to amino acids. So all four of those are secreted from those brush border cells. They're part of the brush border enzymes. Maltase, sucrase, lactase, and dipeptidase. Remember, things that ended ASE are enzymes. And usually the first part of the word will refer to the substrate. So maltase acts on maltase. Sucrase acts on sucrase. Sucrose. And lactase acts on lactose. We will put this in a chart form in the next chapter. Okay, the liver. The liver has some 500 different functions, very important. Receives blood from two vessels in the inferior surface, right? We said we have that triad of structures. We have a duct, an artery, and a vein. So on the undersurface, entering is the hepatic artery carrying oxygenated blood. The hepatic portal vein, remember, is formed when multiple veins from your digestive organs, your stomach, spleen, pancreas, small intestine, large intestine, they're all going to form into one vein and travel to the liver first. That's what forms a portal system. So the blood supply into the liver is both oxygenated blood and deoxygenated blood, hepatic artery and hepatic portal vein. They're traveling to the liver. Traveling away from the liver is going to be the right and left hepatic ducts. Lobes are divided into these hexagonally shaped lobules. They contain plates of hepatocytes. So the hepatocyte is just a term for liver cells. And they radiate out from the central vein, or really from the walls of the periphery of the lobule to the central vein, because that's the direction that blood is going to flow. They have the appearance to radiate outward, but that's not that's not describing the direction of blood flow. The blood flow will be from the outer periphery of the lobule toward the central vein. Then the central veins merge to form the hepatic vein, which then transports blood to the inferior vena cava and back to the right side of the heart. At different corners and walls of these lobules, we have the portal triads. So essentially, it's the same three things that we've identified entering on the inferior, well, entering and exiting on the inferior surface of the liver. We said we have our, triad there, the hepatic triad. Within the lobule, we call it technically a portal triad. So it's composed of one hepatic artery, a hepatic portal vein, and while we're in the liver, we're just going to call it a bile duct. Not a common bile duct, that's going to be farther down. It's just a bile duct. So those three things pal around together. The hepatic portal vein on the slide, they're very, very large. They're kind of like the Grand Canyon of veins compared to their accompanying hepatic artery and bile duct. Those are going to be basically about the same size and sometimes rather difficult to distinguish from each other. But remember, arteries have a muscular tunica media and will have blood in the lumen. Often you'll see erythrocytes there. The bile duct does not have a muscular wall, so it often collapses on itself. It's one single layer of simple cuboidal epithelial tissue. And the material in the lumen is bile. It is, you don't have erythrocytes there normally. So that's another way to distinguish between the two. Right, the sinusoids are capillary-like vessels that empty into the central vein. They have hepatocytes, what's called Kupffer cells dispersed through there, but mostly hepatocytes. Remember, capillary is composed of simple squamous epithelium. So we call these capillary-like because blood is passing through there, but they're not lined with simple squamous epithelium. They are lined by hepatocytes. And then the central vein, in the centers of these all unite to form the hepatic vein, which empties into the inferior vena cava. So finally, a picture that is magnified over to the right, but on the left, you can see the repeating nature of these lobules, these hexagonal kind of stop sign looking structures. So when we look at one, we see the central vein to the interior. Those little pathways going from the periphery of that lobule toward the central vein Those are the sinusoids, hepatocytes all through there, blood passing through there from being transported from all the sides of that lobule and then draining toward the center. Where one lobule meets another, you can somewhat see, I mean, this connective tissue can hold them together, but intermittently you can see these larger openings there, and those will be the hepatic veins, and sorry, the hepatic portal vein. And next to it, generally to one end and to the other, will often be a hepatic artery and a bile duct, and then on the other end, there's another hepatic artery and a bile duct, and they're going to be dispersed all around the periphery. And they supply, the two lobules next to those vessels. All right, so here I like this image because it's showing you exactly what's happening or what these triads are composed of. In blue, you have your hepatic portal vein. In red, you have your hepatic artery. In green, you have the bile duct. So blood is, and over to the right-hand side here, it describes in the lower the portal triad. All right, so we have the bile duct in green, the portal vein in hepatic portal vein in blue, and the hepatic artery in red. And it's showing you with arrows the direction of blood flow. So the blood is not only traveling from the artery into the lobule, and each region, you have several portal triads surrounding this lobule. Each is delivering, along with the hepatic portal vein, blood through those sinusoids that then drain into the central vein in the center, and then multiple central veins will merge to form a hepatic vein, and then off to the inferior vena cava you go. So blood is heading toward the central vein in the artery in the vein, and away from this lobule, where the bile is traveling away from, opposite direction from the lobules, that will then merge to form the right and left hepatic duct, which merges with the cystic duct, and we'll look at what's called the biliary tree. So liver functions, it has many different functions, over 500. It processes nutrients from the small intestine. It processes anything absorbed from the stomach, any of the digestive organs. Helps synthesize plasma proteins, detoxifies alcohol and drugs in blood from the stomach and the small intestine, stores glycogen. Remember, this is where we're going to make anti-A and anti-B antibodies. Should we be blood type B or blood type A? We produce bile there. It's an alkaline solution containing bile salts and pigments. Bile also transports cholesterol out of the body, and conjugated bilirubin from the RBC end products. So remember when we were talking about breaking down that pathway, breaking down red blood cells in the spleen where we produce unconjugated bilirubin, and then it's going to pass into the blood because we're going to put molecules around it so we can be transported to the liver. It's going to have to go into blood, in venous blood. So we transport that to the liver. Now we have cells there like the hepatocytes and things that are going to act on that unconjugated bilirubin to form conjugated bilirubin. And that gets put into bile. And that follows the biliary tree in green to be stored and concentrated in the gallbladder, and then eventually passes into the small intestine. So bile contains bile salts and different pigments of, red blood cells. It contains conjugated bilirubin. That's part of the hemi group that we're trying to eliminate. RBC end products. Bile acts as a digestive aid, not an enzyme. It is not an enzyme. Another component of the bile is the ability to oral function, given the molecules there, is that it can emulsify fats. And emulsification means you surround, or rather an insoluble molecule with other molecules that allow it to be soluble. So it's sort of like the phospholipids of a phospholipid bilayer, that the fatty acid tails point to each other, because a phospholipid has both ends that are hydrophobic and hydrophilic, and therefore it forms that fat layer around a cell. Well, now we're eating fats, and so to prevent or to take a large quantity or globule, let's say, of fat, we want to break it down into small little droplets so that when we release the lipase from the pancreas, we can start breaking it down to its building blocks and it can be absorbed across the mucosa of the small intestine. So to do that more effectively, we want to break a large quantity of something into little droplets of it. And that's what, some of these molecules in bile will do, is it will help surround it, right, and break the fat into small quantities so that now the lipase enzyme can act on it and break it down into an absorbable molecule. Okay, the gallbladder is found on the inferior surface of the right liver of the lobe, or the right liver lobe, I should say, the right liver of the lobe, the right lobe of the liver. It functions to store and concentrate and release bile. Bile is produced by the hepatocytes, of the liver, and they produce the bile that passes through the right and left hepatic ducts, that can then kind of back up the cystic duct and be stored in the gallbladder. And from there in the gallbladder, it becomes more concentrated. When needed, the gallbladder will contract and it'll push the bile back down the cystic duct where it merges with what's called the common hepatic duct. And then as it travels forward, it becomes the common bile duct. And that transports all the bile into the pancreas where it merges with the pancreatic duct. That merging point has to dilate, and that's called an ampulla. So it's called the hepatopancreatic ampulla or the ampulla of Vater. And then from there, it has to enter the small intestine. So, regulating the passage of the bile and the pancreatic juices from that ampulla area that's kind of storing it into the small intestine is another sphincter. And that's your sphincter of Oddi or your hepatopancreatic sphincter. The bile duct and the pancreatic duct unite to form the hepatopancreatic ampulla. The ampulla opens into the duodenum. The hepatopancreatic sphincter or sphincter of Oddi controls the entry of bile and pancreatic juice in the duodenum, like any sphincter does, controls the passage of material. It can contract or it can relax. You also have an accessory pancreatic duct superior to your hepatopancreatic ampulla, and that is, direct. That is just for pancreatic juices to travel through. So we're going to look at the picture and this will make more sense. Let's start up in the liver. We have a right lobe and a left lobe of the liver. Transporting bile from the right lobe is the right hepatic duct, and transporting bile from the left lobe is technically the left hepatic duct. They merge like, arteries and veins would do. They merge to form a common hepatic duct. So that's why they are named as such. We have the gallbladder, and the duct from the gallbladder is called the cystic duct. Those two merge and transport bile to the pancreas, where it can merge with a pancreatic duct, a very large pancreatic duct. Where those two structures merge and dilate is called the ampulla or the hepatopancreatic ampulla. And then what regulates the fluid from those two systems passing into the small intestine is the sphincter of Oddi. You can see the little circular muscular opening there, or the hepatopancreatic sphincter. You can see it on the other side as well. But in addition to that pathway, we have a pathway just for the pancreatic juices alone. And that's what's called the accessory pancreatic duct. Here, superior to that, do you see where there's that other duct that connects to the small intestine? So when we don't necessarily want bile, but we just want kind of a steady flow, of some of that pancreatic juice, it has its own route. The pancreas is composed of Pacini cells. And don't forget, we also have the islets of Langerhans, alpha and beta cells that function in the endocrine system. But now we're looking at the rest of the cells. And those are called Pacini cells, and they produce pancreatic juices. The pancreatic juice contains digestive enzymes and a lot of bicarbonate. The enzymes it contains are pancreatic amylase, In the mouth, we had salivary amylase. Now we have pancreatic amylase that does the same thing. It's going to continue the breakdown of polysaccharides to disaccharides, or starch to disaccharide, because starch is a polysaccharide. We also produce trypsin and chymotrypsin that continues the process that started in the stomach, breaking down a polypeptide to a dipeptide. We have carboxypeptidase that cleaves the carboxyl group off the end of an amino acid chain. So at the end of each amino acid chain is a carboxyl group, a COOH group. And we have a particular enzyme that cuts that off and cleaves that off. We have lipase that breaks down the triglycerides to its building block of glycerol and fatty acids. And we have DNAs and RNAs that act on DNA and RNA to break them down to the building blocks of nucleotides. This is all from that chemistry chapter, the organic portion of the chemistry chapter in AMP1. So, basically how, where these enzymes are coming from right now for the quiz on this material, know which organ, produces which enzyme. It will come together by the time we have the next chapter, because we're going to put this, in a chart and look at each organic family: carbohydrates, proteins, lipids, and nucleic acids. What are the enzymes that break down, let's say the macromolecule to the intermediate molecule, and where do they come from? And then the intermediate molecule to the building block, from a polysaccharide to a disaccharide, and from a disaccharide to a monosaccharide. What are those monosaccharides? Again, what are those disaccharides? It will come together. Also from the pancreas, big bicarbonate. Bicarbonate, HCO3, negative charge, neutralizes the gastric secretions. And don't forget, in the pancreas, we have those islets of Langerhans, alpha beta cells that act to produce insulin and glucagon in the endocrine system. Bile is produced by the liver. Bicarbonate is produced by the pancreas. Okay, the large intestine, the gross anatomy includes house drum. These are these sacs formed by smooth muscle bands. Looks like you took little tucks in that large intestine. We have subdivisions of the cecum that lies below the ileocecal valve, which contains the appendix at the inferior aspect. And then we have the colon that is broken into different regions: the ascending colon, transverse colon, descending colon, sigmoid colon, and then it terminates in the rectum. It has the anal sphincter at the end to regulate defecation. So here we have the large intestine, where the ileum meets the large intestine is that ileocecal valve. Ileocecal valve. So the pouch inferior to that is called the cecum, and the smallest extension there is the appendix. Then you travel upward. You have the ascending colon, transverse colon, descending colon, sigmoid colon, kind of S-shaped and heads back toward your sacrum. And then you have the vertical terminal region of the rectum with the external anal sphincter to control defecation. You see how you have these little rings or tucks that are in the large intestine. Each one of those little regions, from muscle connective tissue at each interval, is one house drum. And the type of physical movement that occurs from house to house drum is called hausdorff churning. So the large intestine is very little breakdown of food. It's pretty much, it is enzymatically broken down and 99% absorbed, if not more, in the small intestine. It's where we absorb most water, is in the small intestine. By the time we get to the large intestine, we're producing and absorbing some vitamin B and K. We reabsorb some water, some electrolytes, and fat-soluble vitamins, A, D, E, and K. But the major function or job is to form feces. Yes, there is some vitamins, that are made by some of the bacteria that reside there, like E. coli. But the major job is to store four pieces. Hormones of digestion. We're going to look at three hormones: gastrin, secretin, and CCK. Gastrin is produced by the stomach and acts on the stomach. The stimulus for the release is food in the stomach or elevating pH of the stomach. And that tells the stomach, okay, we've got food here. We need to start releasing these secretions that the stomach produces, different cells, to facilitate digestion. So it stimulates hydrochloric acid release in the stomach, which converts pepsin into pepsin to begin protein digestion. It stimulates gastric churning. It stimulates cells to release intrinsic factors, so forth and so on. Secretin and CCK are produced in the small intestine. And again, just like I did with some of the hormones, they somewhat have overlapping-ish functions to a certain extent, because once you release that and you relax the sphincter of Oddi, you're allowing bile and pancreatic juices, to flood into the small intestine, but the reasons for them or the stimulus a little bit different. The stimulus for secretin is mostly responding to the acidic environment now in the small intestine, because that chyme from the small intestine, from the stomach, is entering the small intestine through the pyloric sphincter. So, okay, we're starting to get that chyme entering the small intestine. What should we do? Well, we need to neutralize that, right? We need to get some bicarbonate from the pancreas entering the small intestine. All right, so that very acidic environment stimulates secretin, right, to target the pancreas and relax the sphincter of Oddi, or the hepatopancreatic sphincter, so bicarbonate, right, can enter into the small intestine and to start elevating the pH of the chyme, right, to neutralize it. It also feeds back and temporarily kind of shuts off gastrin so that it stops secreting some hydrochloric acid for a while and all those secretions, and kind of let the small intestine catch up with what's there. And then once the pH is modified, you stop secreting secretin. If you're still eating, then we will stimulate more gastrin to be released, continue to digest what's there, and so they kind of go back and forth, stimulating and inhibiting one another, so to speak. All right, then we have CCK. The primary stimulus is fat and even amino acids, some of the proteins that are in the chyme, and that should be CHYME, not CHIME, that's entering into the duodenum. So now we're going, okay, we need to get some bile and pancreatic juices, really flowing into the small intestine, because we need to emulsify the fats and start enzymatically breaking it down, and the proteins and other things that are with it. Right? So it targets hepatopancreatic ampulla and sphincter to relax. It'll target the gallbladder to contract and push down a lot of bile also, so that we deliver a lot of enzymes in bile into the small intestine. Okay, when it comes to chemical digestion of food, this occurs via hydrolysis. We must digest all our foods into the simplest unit for absorption. And so, when we looked at the chemistry in the organic chemistry portion of A&P1, like the second chapter, we ended up looking at not only the inorganic things, but the organic families of carbohydrates, proteins, lipids, and nucleic acids. And we looked at the building blocks, the intermediate molecules and the macromolecules. Well, it's important to understand that those building blocks are the molecules that we can absorb. Anything larger than that, we can't absorb them. So the carbohydrates need to be broken down into monosaccharides. The proteins need to be broken down into the amino acids. The lipids, which, the basic, most common lipid is a triglyceride, needs to be broken down into its fatty acids and glycerol. and the nucleic acids need to be broken down to the nucleotides. So this begins chemical digestion. I'm going to take a break from here to kind of stop, and then we're going to have another Zoom that continues to the chemical digestion, because there's a good amount in there. And the lab, one of the labs is just about the physical anatomy and function and histology of all those structures along the alimentary canal. And then the chemical digestion is a subsequent lab. So I'm going to break here, and then I'm going to make a Zoom for just the chemical digestion.